Hello, we're glad you joined us for this live webinar, Obtaining Samples for Precision Medicine Studies, an Outsider's Perspective. I am Christina Mahalik of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. To learn more, visit labroots.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them in the Q&A box, which will open when you click on the green Q&A button at the lower left of the presentation window. Questions will be answered at the end of the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at the lower left of the screen. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of the webpage and follow the process of obtaining your credits. It is now my pleasure to introduce today's speaker, Matt Klusas, Chief Commercial Officer, OmniSeek. Matt Klusas serves as Chief Commercial Officer at OmniSeek. He leads the OmniSeek sales, marketing, business development, and customer service functions. Prior to joining OmniSeek, Matt was most recently at Thermo Fisher Scientific, where he led a business focusing on large-scale deployments of genetic analysis technologies, including next-generation DNA sequencing, capillary electrophoresis sequencing, qPCR, and other technologies. Prior to Thermo Fisher Scientific, Matt held management roles at the American Type Culture Collection, Life Technologies, and McKinsey & Co. Matt is published in the Journal of Commercial Biotechnology and the McKinsey Quarterly and the Credit Union Journal. In 2015, Illinois State University selected him the Young Alumni of the Year. He received an MSc in Economic History from the London School of Economics and a BS in Communication from the Illinois State University. He also studied at Magdalen College, University of Oxford. Mr. Klusas will now begin his presentation. The floor is yours.
Thank you very much. It's a pleasure to join you today. Again, my name is Matt Klusas. I'm the Chief Commercial Officer at OmniSeek. A quick disclaimer before we begin. The opinions expressed here represent my own and not those of OmniSeek. There's some additional disclosure language there in the bottom right-hand corner of this slide if you need more information. I'm very pleased today to speak to you about a challenge for precision medicine that I think is very present and very important for the field to address over the coming years if we're to realize the vision of precision medicine that we hold as an industry. So today what I'll be speaking to you about is obtaining samples for precision medicine studies. I've called it an outsider's perspective because I'm relatively new to this challenge of trying to obtain samples, and many of these insights are from my perspective as someone who has experience in the life science tools space, as well as someone who has experience recently in the clinical space. So with that, we'll go ahead and move into a brief introduction of myself. I appreciated the introduction just a few moments ago. I've really been in two industries in my career, financial services and life science tools, and more recently at OmniSeek, really in the clinical molecular diagnostics space. You can see a host of different institutions I've been with on the left-hand side. Please forgive the photograph. It's probably the best I can do, so that's okay. A little information about OmniSeq on the right-hand side. OmniSeq is a subsidiary of the Roswell Park Cancer Institute in Buffalo, New York. Roswell Park was the United States' first comprehensive cancer institute. The organization is essentially a spin-out of the Molecular Pathology Lab. We offer a variety of New York State CLEP-approved assays, both next-generation sequencing, CE, and other platforms. The company's mission is very much aligned with the broad goal of precision medicine. Today, that is to try to find the right drug or the right trial for every patient. And as we go through the rest of this presentation, I think you'll see how our endeavors are really aligned with the challenges that we're discussing today for precision medicine moving forward. I want to start off by discussing some of the different objectives of life science tool companies versus clinical companies. And this will specifically be in the space of molecular diagnostics. So now that I have experience in both of these arenas, it's very interesting to compare and contrast where these organizations line up and certainly where they differ. So on the left-hand side, the main purpose of a life science tools company is to produce instruments and reagents that can be used by the community for both research and clinical purposes. The clinical market in a lot of these cases is relatively nascent, especially on the next generation DNA sequencing side. Other sides like mass spectrometry, qPCR, real-time PCR, these technologies have been in the clinic for some time, but sequencing is truly a revolution that has already swept through the research arena and is now making its way to the clinic. These companies also focus on certain regulatory markings such as FDA approval in the United States, CE-IVD approval in Europe, and other markings in China and other countries. Primarily, though, I think one of the main differences from the next segment we'll talk about is that these companies are focused on businesses to produce these materials, distribute them, and ensure customer satisfaction. And so they are organized to that purpose. On the flip side, as we look at clinical companies, they have different objectives in mind. So first, for any assay that a molecular diagnostics company produces, they typically have to demonstrate analytical validity, clinical validity, which is demonstrated by the two subpoints I have there, a retrospective study and a prospective clinical trial. And then the ultimate goal is to be incorporated into guidelines such as NCCN and others that ultimately lead to these assays being included in the standard of care. Now, I've drawn two arrows on this slide because I do think there are some similarities but yet some differences between these. So analytical validity for clinical companies is really demonstrating that the technology and the assay itself is doing what it is that the company is claiming it does. That is very similar to what life science tool companies have to do in terms of ensuring to the market that their platforms are accurate and their reagents are robustly manufactured in a consistent fashion. However, there is a slight variation there that I think you can see. And then secondly, for clinical validation, this is similar to an FDA approval or a CE-IVD marking. However, the hurdles to go through that process are different. Typically for FDA approval, for example, you'll go ahead and get the platform approved with one assay in particular in mind, and then the FDA approvals can follow from that. CE-IVD markings in Europe have a slightly lower bar, which is why many life science tool companies start with CE-IVD approvals and then move from there. But it is very similar on the clinical space to the clinical validation, but the robustness of the study has a different focus and a different rigor to it for clinical companies. Moving on, I'd like to just very briefly talk to you about the experience that I'll speak to here, and that was our efforts at OmniSeq to develop a retrospective study for an assay we're calling OmniSeq Immune Advance. As many of you probably know, the immune checkpoint and immunotherapy space in cancer is revolutionary in terms of patient care, as opposed to targeted therapy where you're talking about responses that are perhaps in some cases several, 6, 12 months relative to a chemotherapy treatment on a Kaplan-Meier curve. With these immunotherapies, we're actually talking about durable responses. And that is a very breathtaking advancement for the field. So this test uses 5 test modalities. You can see them on the right-hand side. I won't walk through all of them, but what we have been trying to do at OmniSeq is develop a retrospective study to validate this assay with the proper guidelines and authorities. Moving on, let's really get into the meat here in terms of the topic that I'd like to speak about today. These retrospective studies that are required for molecular diagnostic companies to go ahead and consider doing a prospective clinical trial, they require a lot of samples. To get a statistically significant population, you typically need to go beyond one institution. Many companies will have a good relationship with one institution. They may be a subsidiary or may have an investor that actually is a healthcare provider, whether this is a cancer center, it could be a comprehensive payer provider network. There are other means by which companies will typically at least have a relationship with one healthcare system. Depending upon the size of that healthcare system, though, it may be necessary to go beyond that institution in order to actually fill out the required samples for a retrospective study. Now, the number of variables will have a resulting impact on the size of the study that you need to conduct. The typical rule of thumb is the more variables that the assay is interrogating, the larger the sample size needs to be in the retrospective study. So if you're only interrogating one variable, the number of samples in terms of a statistically significant standard that journals would be happy to accept will be a little bit lower than if you have multiple variables. The ideal study then will typically include anywhere from several hundred to potentially thousands of samples. And there are examples of successful molecular diagnostic companies that frankly never stop building their population in a retrospective sense. You can get into semantics about whether once the assay is released it's really a prospective addition, but nevertheless it's really around collecting the proper reference population to essentially validate the use of the assay moving forward. One of the biggest challenges, and I think this is really where the rubber meets the road on this issue, is the significant attrition that can be expected as one goes about trying to obtain samples for a retrospective study. In our experience and in the experience of others that I've anecdotally surveyed on this topic, the attrition rate for actually pulling in retrospective samples is very significant. And that's not only within a single institution, but as you look across multiple institutions, you can expect anywhere from 80% plus attrition from the number of samples that are initially identified down to the actual number of samples that qualify for the study that you're looking to conduct. And so that in and of itself creates a significant challenge to getting these types of studies done, and then by extension actually producing new innovations in the field of precision medicine. So some of the impediments that lead to this significant attrition or possibly just a challenge in terms of obtaining samples at all really cut across all of the different types of institutions that you're likely to reach out to obtain these types of samples. I've listed 3 here, and we'll get into some more on a segment-by-segment basis in just a moment. But let's focus on these first 3. So the first is price per sample. This is obviously a budget constraint for many organizations. The challenge with molecular diagnostics is that you're typically competing with pharmaceutical companies. These companies will tend to spend anywhere from 5 to 10, perhaps even more, on a per sample basis for qualified samples that would go into a prospective clinical trial. As you think about the number of samples required for a molecular diagnostic assay, for most organizations that number is going to be too high simply because the economics of molecular diagnostics is so much different than pharma. Just for an example, the lifetime value of a pharmaceutical customer could easily be in the 6 figures, whereas for a molecular diagnostic company, the lifetime value of a customer is probably going to be in the low 5-figure range. And that's assuming that this provider is going to be ordering a lot of tests over a several-year time span. And so the differences in those economics, I think, really then feed into the budgets that a molecular diagnostic company can offer relative to a pharmaceutical company. Labor availability is the next significant challenge. When conducting a retrospective study, there are 2 components that really come into play. First is identifying, pulling, preparing, and shipping the samples themselves. And the second component is around collecting the data that goes with those samples that will then feed into the study. So that's really 2 different we'll call them labor pools that need to be tapped for obtaining samples for a retrospective study. In doing so, you tend to touch a lot of different people within these types of institutions, and I've listed some examples here on the slide. So oncologists almost always have to be involved, but depending upon the nature of the study, you may be able to get away with a research fellow or someone of that nature. Radiologists are typically required insofar as for rhesus criteria, you typically rely on scans to measure disease progression. And so getting time from those folks is also important. As I mentioned, in terms of pulling samples, you'll typically have operations folks that are in banks that you'll need to work with in terms of actually pulling the samples themselves, preparing them for shipment, making sure that you're either obtaining the entire block, which is typically not the case, or you're obtaining a number of cut slides of various numbers to go ahead and have those sent over for your study. And then finally, you're competing with the day-to-day operations of the institution themselves and perhaps competing studies. Those competing studies can be investigator-initiated studies within the institution itself. They can be pharmaceutical clinical trials. It really varies. And then, everybody's busy on a day-to-day basis. So this tends to be, to a certain extent, incremental work for these institutions. So labor is really a key challenge. And we'll talk a little bit later about some ways to ensure that you are not asking too much on the labor front that would actually put your ability to obtain samples at risk. Finally, across all institutions is an IRB process. This question, while it may not emerge in the initial conversation, in my experience, I would be comfortable saying that almost in every case it will come up at some point. The interesting thing about this is that for a non-interventional retrospective study, which is about the lowest bar you can do in terms of an IRB approval, the necessity for having an independent IRB verification, accepting other institutions' IRB approvals, formatting your own for the specific institution that may contribute samples, there tends to be amazing variability between how institutions interpret the IRB process relative to a study of this nature. So as you progress, and again, we'll talk a little bit later about some of the strategies to ensure success, this is an area where flexibility is really key. And that's the point I've called out here at the bottom. Across all three of these components, one size does not fit all. The price per sample, can vary significantly between institutions. Labor availability, depending upon the nature of the institution, will be significantly variable. And again, this IRB process is likely to be variable as well. So it's very important to stay flexible to the institutions that you're dealing with, and instead of trying to impose your process and your understanding of the issue, you really do need to do more listening in these cases to respond to the protocols that they may have set up, the institutional guidelines, and just the general practices of the partnering institutions to make sure that you can obtain the samples that you need. Other impediments are segment specific, and so what I've done here is I've identified 3 different segments that we'll talk about on the next slide, and we'll get specific on each of these. But in our experience, you can really divide up the typical challenges that you encounter with a retrospective study by these 3 segments. The first one are your major academic medical centers. I've been specific here not to list specific names because I certainly think that these issues cut across all institutions within these segments, and I certainly didn't want to put one institution on the spot. So major academic medical centers are one case. There's probably anywhere from, let's say, 10 to 25, maybe 30 of those across the United States, depending upon who you ask. Biobanks are another source. Biobanks can be attached to institutions. They can be independent. There are some kind of third-party biobanks out there that have relationships with other biobanks and can query them to pull together samples. The number of them tends to be more than the major academic medical centers, but I would guess you're probably not talking about more than 40, no more than probably 50 material biobanks out there that could potentially provide samples for a retrospective study. And then finally, you have your mid-tier cancer institutes and your community practices. This is really the bulk of where cancer is practiced across the United States, and I would venture in other regions of the globe as well. These institutions, in terms of number, you're well into the thousands. Mid-tier cancer centers, you're probably looking somewhere in the hundreds. Community practices are well into the thousands. Let me cover the final point on this slide down at the bottom around experience. So when it comes to these types of studies, you will see a lot of variability across these 3 segments. Major academic medical centers tend to be the recipients of a lot of pharmaceutical-sponsored clinical trials. They tend to have a lot of experience with things like rhesus criteria, evaluable scans, tissue in stored and available in the types of way that are required for retrospective studies. They have a lot of experience, and I apologize, I've just realized now I didn't label these Harvey balls. So a full Harvey ball in this case means high, and an open circle or kind of that quarter moon that I have there, that means low in this case. So continuing on the experience, biobanks in our experience don't tend to have, surprisingly, a lot of experience with retrospective studies. And this was surprising because a lot of biobanks will advertise that they are available for these types of studies and they want to do these types of studies. But my hypothesis would be that in most cases they are receiving requests from researchers who are doing research use only studies and don't have enough experience at these times to really be able to respond to to the rigors of a clinical retrospective study. And so it's a little more challenging to get these institutions over the line. And again, we'll talk more about some of the specific issues with biobanks in just a moment. Your mid-tier cancer centers, your community practices, you'll see a wide variety of experience with these. Mid-tier cancer centers will do prospective clinical trials with pharmaceutical companies. Typically not in the numbers you see at the major academic medical centers, but they'll likely have some amount of experience. Some community practices have never attempted something like this in the past. And so you have a lot of variability within that segment in terms of experience. Gauging the experience of a particular institution can be pretty challenging. But there are some probing questions you can ask to get a sense for what they've done in the past, what resources they have access to in order to be able to complete a study of this nature. So it does help to go into those conversations with the thought around probing on their experience so you know exactly what type of institution you're dealing with. Okay, moving on. So there are a number of pros and cons in terms of working with each one of these segments. And I wanted to be sure to call out the pros because although we're talking about challenges with precision medicine, I think it's important to look for the light at the end of the tunnel in these types of studies. And for those of you who have not done them before, I can say comfortably from experience that the process of doing these is very challenging. And there will be a lot of times where it seems like there is not a light at the end of the tunnel. But in our experience, there typically is. It will just take a lot longer than you'd expect. And I'll talk a little bit more about that in just a second. So let's go through this slide. So major academic medical centers, I've already touched on some of these points, but because of their experience with especially pharmaceutical and investigator-initiated clinical trials, they'll typically have very robust data. They'll typically have very well-managed biobanks. They will have a very high interest in new research. So if you're offering a new molecular diagnostic assay that is tapping into something that is very current, that is very quote-unquote sexy at the moment, you should be able to attract quite a bit of interest from these institutions. And finally, a lot of these institutions employ a lot of the key opinion leaders that will be critical for supporting this assay once it reaches the market. And so working with at least one or two of these types of institutions is very helpful because it not only sets the groundwork for the retrospective and the prospective trial, but it also sets the framework for market acceptance once the assay is released. The cons with working with major academic medical centers, I've listed a few here. Probably the biggest one, I would say, in our experience is actually the second bullet under there. The separation between the oncologist, the clinicians, and the pathology department. In some institutions, you'll see these two groups really working in lockstep, no separation between them. In other cases, it seems as though they may be on different planets. And I really do mean this in kind of a joking sense. But you will have, in some cases, a pretty wide gulf between an oncologist or a clinician being very excited to participate in a retrospective study and the motivations and the desire for their pathology colleagues to engage in the study in a similar fashion. That's probably the largest one that we've seen. Another significant one is competing research. And the challenge around this one is that you're not only talking about competing research that's defined and in process, you're also talking at these major academic medical centers about the prospect for future research. So in some cases, with these samples, you're talking about very precious samples that a retrospective study of this nature would go some way, if not all the way, to exhausting the tissue available for future studies. And in some cases, you will encounter researchers who may not have a particular objection to the study you're proposing. They may actually think it's very interesting, but they may not be willing to part with those samples simply because they want to preserve them for a research study that they would like to do sometime in the future. It's a very difficult objection to overcome, but it is part of the reality of dealing with these institutions. Finally, there's the institutional buy-in. Major academic medical centers almost entirely tend to be very large institutions with quite a bit of bureaucracy. It's sometimes hard to find out exactly who the decision makers are, exactly the process for getting approval to engage in a study of this nature. In some cases, that may manifest in multiple phone calls with multiple different audiences. And again, this is where patience really does come into play. Let's talk a little bit about biobanks. So in terms of the pros with biobanks, they are set up in such a way that should make them amenable for retrospective studies. They tend to operate primarily on a fee-for-service basis. So as opposed to with the other two segments where you're not only trying to offer some kind of fee-for-service arrangement, but you're also trying to get potential collaborators excited about what you're doing and get them involved in the study itself. With biobanks, you kind of throw that out the window. You just focus on the fee-for-service notion and you move from that point. Because of that, biobanks do have the potential to move quickly in terms of providing these samples and getting them out. The economics do tend to drive this segment, so the old mantra of cash is king does tend to come into play. However, as we kind of transition to the cons, the precise requests for the samples themselves does tend to play a pretty significant role in the response that you'll get from biobanks. If it's an easy ask for the biobank in terms of both the samples and the data that you're requesting, you can expect pretty quick turnaround. If, for example, though, you're asking for quite a bit of data, you're asking for quite a bit of a sample, that can kind of put the brakes on the speed with which biobanks will operate. You do tend to see a lot of variance within biobanks in terms of the types of materials they have available as well as the data. And the challenge with both of these is that with biobanks you tend to get what you see. There's not a lot of opportunity with most biobanks to go back and, for example, obtain more tissue, which is very rare in any case, but especially on the data side, there's very limited opportunities with biobanks to go back and pull more data, simply because the biobank will typically employ an operational staff, perhaps a business development function, but they don't tend to have relationships in which they can go back to an oncologist, to a radiologist, even to a research fellow, and get additional information that you may need for a study. So that in itself does create a bit of a challenge where if you do need additional information from a biobank, you may be better off continuing your search in other areas, because the likelihood of receiving that information is not terribly high. Finally, in terms of mid-tier cancer institutes, community practices, the nice things about working with those institutions is that they tend to have a more straightforward decision-making process. It's easier to identify who the decision maker is, get that person engaged, and hopefully get the approvals you need pretty quickly. They do also tend to have a very high interest in publishing. These institutions are always looking for more notoriety, more publications that can increase their stature, possibly increase their ability to attract government funding or other kind of charitable donations, and so that is a key motivator for trying to sell these institutions on participating in a retrospective study. On the con side, similar to the biobank, as I mentioned before, you'll see a lot of variability within this segment. So you will encounter some cases where there are very limited additional resources for data collection, especially when you start talking about community practices. One of the biggest challenges is that the day-to-day operation itself tends to overwhelm their ability to take on a project like this. And so, again, I think minimizing your ask up front is essential for getting engagement with these practices. And finally, relative to the other two segments, you will see quite a bit of variability in terms of the potential number of samples that these institutions might be able to contribute to a study. In our experience, you will be surprised by some institutions who you would never guess, and they may have tens to hundreds, maybe even more. In other institutions that you'd expect they'd have a lot, they may only have a dozen or a couple dozen, something like that. So you've got to be prepared for some pretty wide variability in terms of the number of potential samples, and then as we talked about before, it's always important to factor in the attrition rate into that so that you have an idea of how many samples you're likely to end up with at the end of the day. Okay, so what can requesters do to increase success? We've talked about a lot of the challenges. We've talked about some of the pros of dealing with each of these segments, and I've alluded to some points on this slide already, but let's kind of walk through this in terms of what are the things that your institution can do when conducting a retrospective study to maximize the potential, as you see in the center there, that you start getting samples in the door. Because at the end of the day, that's the most important thing. I'm going to actually start in the bottom right-hand portion of this slide with the legal and the economics. I think it's really important before you have your first conversation to internally amongst your institution define what types of legal frameworks you're going to use to manage this retrospective study and to agree on the economic incentives early on. With that said, my point around kind of flexibility also comes into play here. I would advise having a very clear-cut hypothesis within your institution before that first conversation. But I'd also advise revisiting that after your first handful or so of conversations so that you can gauge whether the hypothesis you had before the first one is actually resonating or whether you need to adjust. Holding firm in really any of these cases beyond the kind of bare bones of what you absolutely need, and that could be need in terms of budget costs, need in terms of sample requirements, by and large, that is the worst thing you can do for trying to successfully conduct a retrospective study because, as I mentioned before, one size does not fit all, and you'll end up filtering out the vast majority of potential collaborators if you hold too firm to any superfluous requirements in a study like this. Let's go up to the top left here, so we'll kind of mix it up a bit. I've alluded to this before, but I think it's really important to define not only your ideal criteria for both samples and data, but also to define what I'll call your minimum requirements. On the second bullet here, I've alluded to a psychology technique that they call foot in the door. There's the foot in the door technique and there's the door in the face technique. The foot in the door technique is you start with a minimal ask to kind of get your foot in the door, and then you, as you get a yes to the first one, you incrementally add additional requests in the hope of working up to your ideal. With the door in the face technique, it's pretty much the opposite. You start with the ideal and then you work back from there. So in our experience, the foot in the door technique does tend to work best because you can get institutions more engaged by saying, this is the minimum criteria that we need for participation in this, but if you can do more, we'd love to talk with you about the ideal criteria that we're shooting for. This does create a bit of a challenge on the back end of conducting a retrospective study, in that when you start sacrificing on your ideal criteria, you do start to limit what you can do from a publication standpoint. And this can include things like waterfall charts, Kaplan-Meier curves, things of that nature. Ideally, if you can get as many ideal criteria as possible, you can produce at least a subset of your overall population with those types of charts in such a way that that you can go ahead and have a publication that's robust with a complete reference population, but perhaps just a subset for some of those charts. Let's go down to the bottom left-hand side here, robust documentation. This is another point that I think is really important to stress with retrospective studies. By and large, the institutions that will participate will be interested. They will certainly be incented by the economics, hopefully the science as well. But it's important to bear in mind that the less work you ask of these institutions, the more likely they are to respond to the work that's actually needed to be done. And so in this bullet, it really helps to think through all of the documentation that an institution could request from you before you have that conversation, so that immediately following a teleconference, you can go ahead and send them a document with everything that they've requested already laid out. What you're doing in that is you're reducing the amount of work that they need to complete themselves, and in doing so, you're allowing them to essentially make their lives a lot easier, which will ultimately make your life easier. So the second bullet on this, I think, is important. I think in our experience, what you want to do is you want to provide some documentation on the study design, on the science, on the ideal parameters for samples, on the economics, etc. You do want to provide that up front, because you have to give potential collaborators some indication of what it is you're working on. But on the back end, as you start to get into protocols, as you start to get into IRB documentation, as you start to get into sample preparation documentation, in our experience, it's typically better to hold that back and to distribute that as it's requested. If you start off kind of sending the entire bolus of all this documentation, again, the human psychology element does come into play here. You risk scaring them off from actually participating with you in the study. And so that's where that recommendation comes in. Finally, we'll go up to the top right-hand portion of this slide, practicing your pitch. So the conversation does take a while to hone. It's best to do a few kind of internal, we'll call them dry runs, so to speak, of your pitch before you have your first conversation with someone external. With that said, you should really look at your pitch as a living, breathing presentation. Some messaging you'll probably find yourself repeating over and over again because it's effective. Others you'll just throw out and you'll put in something new from scratch because either it didn't address a point that you've gotten repeated questions on, it was done so in a way that was too complex for people to understand. I mean, there's a wide variety. So the second bullet I have on here I think is really important to bear in mind. Keep experimenting with different approaches so that over time you will find yourself digging into a pretty comfortable groove in which you have answers to the most frequent questions. You're presenting the material in a way that's accessible for the audience to receive, and you're addressing most, if not all, of the common questions in the presentation itself so that you can really then skip ahead to the questions that are going to be specific to an institution. So all of this said, this is really our experience so far with retrospective studies. They are incredibly important. They are really a key to enabling precision medicine, because without them, it's difficult to prove the utility of a lot of these great innovations that we've developed. And so for everyone kind of listening to this, I would just make a plea that either when you're approached to participate in a study of this nature, or when you're considering one yourself, I think it's important for us as an industry to really try to work together as best as possible to make these things a reality. Because if everyone kind of stays in their own silo, if everyone is inflexible to the potential absolute requirements of other parties, then we'll continue to exist in this kind of space where not as much research is getting done that could potentially get done. And that's really a shame, not only for the industry, but also for the promise of precision medicine as a whole. And finally, my last comment here, this is a picture of my daughter. She's about to turn 2 years old here in April. I think one of the great things about working in this industry is that we really are working on something that will hopefully impact our loved ones, if not ourselves. And so as I think about challenges like these, they're very tactical, but at the same time I'm always considering how success at very tactical things like obtaining samples for a retrospective study will ultimately lead to the breakthroughs that could potentially save the lives of those we love. And what could be more important than that? I thank you for the time. I'm going to go ahead and hand it back off now, and I welcome your questions. Thank you.
Thank you, Mr. Klusas, for that informative presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Mr. Klusas will answer as many questions as time permits. Without hesitation, let's hop in and get to our first question of the day. This question asks, why does the attrition rate for samples tend to be so high?
Yeah, that's a great question. I mean, you would think that identifying the potential cases, I mean, let's just say, for example, in a retrospective study, you're looking for patients that have been treated with a particular drug who are above 18 years old and have not had their first dose of the drug yet for the sample. You would think that close to 100% of those samples would be available. The challenge is really when you get into two things. One is the data requirements, and secondly is the samples. So even though pharmacy records at an institution may indicate, let's say 400 patients have been treated with a particular drug, once you go in and you say, well, who actually has samples that can be pulled and analyzed, you're probably going to have a pretty significant drop-off just at that point. And that could be because the initial sample was very small, such as a fine needle aspirate, or it could just be because they sent the sample off somewhere else for analysis. It may be with another lab. There are a whole host of reasons why that can happen. That's really the first reason. The second reason is around the data requirements. As you start looking at who has valuable scans for doing things like waterfall plots, as you start looking at outcomes data, institutions may be a second opinion institution, so they may not have actually followed up with that patient after the initial treatment. In other cases, they may have, simply not tracked that person after they stopped coming in, even if they were in a local setting. So you do tend to see a wide variety of reasons why the attrition rate is so high, and really what it is, it's a compounding factor across all those different variables that leads to that 80% plus attrition rate.
Thank you so much for that answer. Our second question of the day asks, how is your organization dealing with these challenges to advance precision medicine?
Yeah, thanks. That's a great question. The biggest thing I think we're trying to do, and I would recommend this to everyone trying to do one of these studies, with very high attrition rates, with a variety of different challenges with different types of institutions. My boss likes to say sometimes you've got to try to get as many shots on goal. This is a hockey analogy. We're based in Buffalo, so that's probably appropriate. You try to get as many shots on goal as you can. Even if your actual, let's call it a field goal percentage in basketball parlance, even if it isn't terribly high, if you have a lot of shots on goal, if you have a lot of lines in the water, to use a fishing analogy, the hope is that you'll get at least some percentage of those to actually come through. And so what we've done is we've really tried to cast as wide a net as possible to be able to get as many institutions into the funnel as we can, and then we kind of let this natural attrition process take place, and we get the institutions that we have as we get all the way through.
Thank you for that answer. It looks like we have one final question of the day. And this question asks, what are the typical prices on a per sample basis in your experience?
Yeah, that's a good question as well, and unfortunately it's highly variable. So as I mentioned in the presentation, typical pharmaceutical rates per sample tend to be in kind of the $5,000 to $10,000 range. For a retrospective study, I'll tell you, we started offering about $300 per sample, hoping that that would be enough. It turns out it really wasn't. What we tend to find is that most institutions are more comfortable in kind of the $750 to $1,500 range per sample. One thing to bear in mind with this is that some institutions are actually either going outside of their own four walls to get this work done, or there's some sort of pitch and catch internally within the institution where one department has to send funds to another department to get this type of work done. And so in a lot of cases, it's not necessarily greed of the institution that's driving the price, although there is certainly a little bit of that. But it does tend to be related to the rates that are standard either within the institution that you're working with or with their external partners. So we found generally that for a retrospective study where it's a non-interventional project, somewhere in the $750 to $1,500 range tends to be enough to get most institutions over the line.
I would like to once again thank Mr. Klusas for his presentation today. It was truly enjoyable and very informative. I'd also like to thank the audience for joining us today in LabRoots for today's educational webcast. This webcast can be viewed on demand through May 2017. LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. This is Christina from LabRoots, and we'll see you here next time. Goodbye.